Hello everyone, this is Rubina Kausar from the Department of Pharmacognosy. Today I am going to tell you about plant tissue culture. So plant tissue culture is something which we have studied earlier, but how it is applied in pharmacognosy is what we are going to learn today. So before getting into the details, so let me tell you that basically tissue culture is divided into two types. First, plant tissue culture, and the second one is animal tissue culture. But we are not bothered about animal tissue culture here. And now with a small example, with the help of a diagram, today I am going to tell you what is plant tissue culture. So here, first, let me take a small example. So let me draw a diagram here. So before getting into the details, let me tell you a small example here by drawing a diagram. So here I am going to draw or a plant, whatever you consider. So let us assume that this is a plant. So what I am going to do is, here I am talking about conventional growth of a plant. So conventional growth is nothing but how a plant grows naturally. Okay. So please tell me, what does a plant require in order to grow properly? Yes, what does it require? Yes, it is sunlight, next air, next water, next soil, okay, which water is nothing but which comes from the soil, next other essential requirements. What are these requirements? They can be carbon, nitrogen, etc. Yes, apart from this, it also requires certain nutrients. Okay, we'll learn all these nutrients in the later parts of the class. So what I'm talking is all about the conventional or the natural growth of plant. So in a natural environment, if you see, a plant requires all these basic things. First, water, nutrients and other requirements like carbon, nitrogen, whatever it is, they are all absorbed through the soil through roots, and it ultimately reaches the different parts which are present aerially. And apart from these, whatever is coming from the soil, it also requires, plant also requires sunlight and as well as air, right? So this was all about naturally occurring process. So what I'm going to do in plant tissue culture is, I'm going to take, suppose if I consider this as leaf, so I'm going to take a part of the leaf which I call it as explant. So I'm going to consider a small part of a plant, and then I'm going to place it in some nutrient medium. I'm going to place this explant in the nutrient medium and then wait for it to grow. This process is occurring without the presence of soil. I'm not going to add any soil to it. I'm simply taking a part of the leaf which I'm calling it as an explant and then simply placing it in the nutrient medium. Why am I calling it as nutrient medium? Because nutrient medium is something which is full of nutrients. So what are those nutrients? I'll let you know later. So, and one more important thing, why am I calling it as explant? Explant is something which was earlier a part of the plant, That's the reason why I'm calling it as X. X is something which was part, which was, okay, past tense, which is not now a part of that particular plant. And that is supposed to be in the nutrient medium. So in this nutrient medium, this explant is going to get converted into callus, which is a mass-like structure, and then ultimately it grows into a whole developed plant. That all process we are going to study later. And what I was talking about, this nutrient medium, this whole process occurs in the laboratory. So this I am going to carrying it out in laboratory conditions, that is in vitro conditions. In vitro is something in laboratory. So right now you must have understood what is the basic concept behind this plant tissue culture, isn't it? So based upon this, I am going to define the plant tissue culture. So plant tissue culture is an in vitro technique wherein you are going to use a part of a plant. The part of the plant can be either from the root, from the shoot, from the leaves, from the anther, pollen grains, stamen, any part, okay. That part is nothing but the explant, right? So it is an in vitro technique wherein you are taking the part of a plant and then you are providing suitable conditions. That is, In vitro like laboratory conditions, you're artificially providing all these conditions and letting it to grow into a whole new plant in order to produce primary or secondary metabolites.